Hello, my dear friends. In one of my earlier video lectures, I explained you the composition and functions of the human blood. What is blood? The definition of the blood. Blood is a tissue. Blood is a connective tissue. Blood is a fluid type of tissue. It can flow from one place to another place within the body. The main constituents of the blood are blood cells or blood corpuscles and plasma. Plasma is the matrix, a liquid part, whereas the blood corpuscles are three types. Red blood cells RBC or erythrocytes, WBC or leukocytes, white blood cells, white blood corpuscles, the third type thrombocytes or platelets. Now with this background, let us try to understand the blood groups or blood types. If you take all the human beings, the general composition of the blood is same. General composition. The blood cells, the plasma, the constituents of the plasma, but immunologically, genetically, there are different types. Then how to transfuse the blood? Now let us see. So this is Karl Landsteiner, a great surgeon, Austrian immunologist. Karl Landsteiner first discovered the ABO blood group system in 1901. He identified that in all the human beings, if we put all of them blood group wise, they can be divided into four types. It was discovered in 1901. After almost 30 years, his work was recognized, the importance of the work, applications of his work was recognized, and he was awarded the Nobel Prize in Physiology and Medicine. In 1940, he discovered another type of blood group that is known as RH blood groups. This is a great scientist, Karl Landsteiner. Now, let us try to understand what is the difference between transfusion and infusion. Transfusion is the transfer of blood from one person to another person, healthy to patient or healthy to injured. Blood or blood components, sometimes they separate the blood components just like the blood platelets and give it to other persons, needy persons, from one individual to another. Infusion is the introduction of fluid part of the fluid, any fluid other than blood. Remember, other than blood, for example, saline, some salts, or some other things are given, saline solution or glucose. Blood group, I told you, there are four types of blood groups. People can be divided, categorized to four types depending upon their blood type, A, B, AB, O. Now, blood group is determined by an antigen. They are called agglutinogens. Antigens or agglutinogens. These antigens are present on the surface of RBC. Antigens are present on the surface of the RBC. Antibodies, also known as agglutinins, can bind to RBC antigens. Agglutinins bind to agglutinogens or antibodies bind to antigens, we can say like that. Resulting, suppose an antibody binds with antigen, corresponding antigen, there will be an agglutination. That's why they are called agglutinogens, agglutinins. Or what is agglutination? Clumping of the blood. That will result in hemolysis or rupture of RBC. When the RBC rupture, all the contents of the RBC come out. That is a dangerous situation. Then blood groups are mainly two types. One system is known as ABO, another is RH. Both are important, but most important is ABO. Then why the blood grouping, blood typing is important? Nowadays, blood transfusion has become a very common practice in all the hospitals, in all the health institutions. When the blood transfusion is practiced, a person needs, just like a patient, an injured person in accident or in the battlefield, or one who suffers from genetic blood disorders, or in case of surgeries, the people need the blood. In that case, donating the saved blood, transfusion of the saved blood is very important. This safeness is determined by the blood group system. We cannot give every type of blood to every type of person. In fact, it is a dangerous thing. To prevent the hemolytic diseases or newborn RH compatibility newborn, some are born with hemolytic disorders, they need the transfusion of the blood. To solve the legal disputes, suppose a baby is born, two fathers are claiming or two mothers are claiming that the child is mine. In that case, through the blood analysis, one can determine who is the actual mother, who is the actual father, and who are the parents. To study the Mendelian laws of inheritance, how this blood group system is inherited from parents to offsprings, from one generation to another generation. Blood groups, antigens. The blood group system, ABO group or even RH system, is determined by antigens. Antigens, antibody generators. The expanded form is, what is antigen? Antibody generator. Blood group will depend on which antigens are present on the surface of the RBC of an individual. You take an individual, his RBC. On the surface of RBC, some antigens will be present that will decide to which type of blood group he belongs. Antigens present on the surface of the RBC. Scientists most frequently use two types of antigens to classify the blood types. One is ABO antigens and other is RH antigens. ABO is the best known system for grouping of blood groups, most common ABO system. The minor groups are Kell, Kidd, Duffy, etc. They are not so important. In special cases, they assume the importance. Karl Landsteiner, who actually first discovered the four types of blood groups, he proposed almost a theory, a law, which is applicable even today. It is known by, that law is known by Karl Landsteiner's law. Now, what is that law? If antigens are present on the surface of RBC, if one type of antigen is present on the surface of the RBC, the corresponding antibody must be absent in the plasma, his plasma. You take the blood of one individual, on the surface of his RBC there is an antigen. The corresponding antibody will be absent in his plasma. Similarly, if a certain antigen is absent on the surface of RBC, the corresponding antibody must be present. Opposite to this, suppose once RBC is devoid of antigen on its surface, in that case antibody will be present in his plasma. So this you must clearly understand. If antigen is present on the RBC, corresponding antibody will be absent in his plasma. If antigen is absent, antibody will be present in the plasma. That is Landsteiner's law. Now, ABO locus has three main allele forms, ABO. These three types of alleles decide the blood groups. A and B genes found on chromosome 9. Remember, important point. These genes deciding the blood types are located on chromosome 9 and are inherited one gene from parent. Suppose one is AB blood group. A comes from mother, B comes from the father. One gene from father and one gene from mother. Mendelian inheritance. If such is the case, alleles are present, they will be inherited as per the Mendelian's laws. Now, if you take the four blood groups percentage, O blood group people, A, B, AB, in our population throughout the world, most common people are O group, 47. They constitute 47% of the total population. Then A group, 41. Most common blood groups are O group. Later on, A, B, 9, rare group is AB, only 3%. Among the four, the rare blood group is 3, most common one is O group. Then, red blood cells membranes have antigens on the surface of the RBC. Antigens are present. What are these antigens? Proteins or glycoproteins, because they are associated with certain carbohydrates, glycoprotein on the external surface. These antigens present on the surface of the RBC are unique to individual. If my blood contains A antigens on the surface of the RBC, that is unique to me. Recognized as foreign if transfused into another individual. If my blood is transfused, suppose my blood group is A blood group, if it is transferred to another person, the another person recognizes these antigens as A. Promote agglutination of red cell cells if combined with corresponding antibody. I am using the word corresponding. If A antigen comes in contact with the A antibody, then there will be agglutination, clumping of the blood, hemolysis. ABO blood group system. ABO blood group system classifies the blood group types according to the different types of antigens on the blood cells, RBC, and antibodies in the plasma. Antigens on the RBC, antibodies in the plasma. There are four: group A, group B, group AB, group O. Now let us go further into the details. A group people, Suppose one individual blood group is identified as A. He is a blood group person. The surface of his RBC contains A antigen and the plasma has anti-B. If A antigen is present, antibody B will be present in the plasma but not anti-A antibody. Similarly, B blood group individuals, the surface of the RBC contains B antigen, and in that case, their plasma contains A antibody, but not B antibody. Opposite, A antigen, B antibody, B antigen, A antibody in individuals. AB, then third group. In these individuals, suppose some people are AB blood group, one individual AB blood group, or is the RBC. On the surface of his RBC, both A antigen, B antigen will be present. Both. Plasma does not contain any antibodies. The surface of the RBC contains A antigen, B antigen, but antibodies are absent in the plasma. So therefore, they can receive the blood from any other three types of people. They are called universal recipients because antibodies are absent. AB blood group, universal recipients. O, the surface of the blood does not contain any AB antigens. O or 0, we can say. RBC does not contain any antigens. But interestingly, both types of antibodies will be present in the plasma. Now, since antigens are not present, these people are universal donors. They can donate, give the blood to any type of person. Their blood is safe. Therefore, they are universal donors. AB, universal recipients. O group, universal donors. Then, look at this diagram. A blood group people, RBC, on the surface antigen is present. In plasma, anti-B is present. Now, group B, B antigen, see the difference between these two. B antigen, anti-A in plasma, antibodies. Antibodies are anti-A. Then AB, both. See, both antigens are present. Then no antibodies in his plasma. Then O group, no antigens. But both types of antibodies will be present. Anti-A, anti-B. Here anti means antibody. So these are antigens. So this is again very useful diagram to understand. Then let us see what is antigen structure. So diagrammatically, it is very much enlarged. RBC, red blood corpuscles. On the surface of the RBC, antigen is present. Antigen is enlarged and shown the details. Here to the surface attached glucose, galactose, N-acetyl glucosamine, galactose, fucose. Antigen, that's why it is called glycoprotein. Protein and associated with certain carbohydrates. Now, if the structure is up to this, then it will be called as antigen, H antigen. That means H antigen consists of glucose, galactose, N-acetyl glucosamine, fucose, and galactose. It is common, this up to this structure is common for both A antigen, B antigen. Where does the difference lie? The difference here is, suppose if galactose is attached with D-galactose, then that person will become B blood type. It is B antigen, like this. Here, it is also. Not this, but to this galactose, N-acetyl galactosamine is attached. It is A antigen. A antigen means all these are common, this common, this common, and this, but not this. For B antigen, this is not present, but this will be present. This is the antigenic structure. Remember, H antigen is common for both A and B blood groups. A and B. Then, if you take the ABO system, blood group system, A group, B group, AB group, O group, who can give the blood to whom? This is a very simple and very useful informative diagram. O group people can give the blood to A, B, AB. Arrow mark shows the direction in which it can be donated. O group can give to his own blood group, O, A group, B group, AB group. So therefore, he is known as universal donors. O blood group people are known as universal donors. Whereas AB blood group people, you can see, they can receive the blood from A, B, O, and his own blood group AB, or from all the people. Therefore, they are known as universal recipients. Now, you can see, suppose one person is A blood group, from whom he can take the blood. He can take the blood, his own blood group, O blood group. But he cannot take the blood from AB. Similarly, if you take the B blood group, he can take the blood, receive the blood from O and AB, but not from A, but not from AB. The direction indicates, this arrow marks indicate a direction in which the blood can be donated. Transfusion only in the direction of arrow, but not in reverse direction. Then another blood group, Bombay blood group. This is a special type of blood group detected in 1952 in Bombay population. Frequency estimated to be about 1 in 7,600. Very special, only in Bombay. That's why it is referred as Bombay blood group. There will be no antigens, HAB antigens. In Bombay blood group people, no antigens on the surface of the RBC. So, they cannot take the blood either from the A, either from the B, either from the AB or O, but only from their own blood group, Bombay blood group. This is the specialty. No antigens. Their blood is not compatible with the ABO system.